Okay, this next video we're going to talk about some of the causes of cell injury, and then we're going to get into the morphologic features of cell injury and some features of necrosis. So what are the different things that can cause cell injury? So there's a lot of different things, and we're going to go through each one individually. So here's the list, if you want to take a peek at that, and then we're going to jump into each one separately. So the first one is oxygen deprivation. So hypoxia is a deficiency of oxygen, and so when a tissue or a cell does not have oxygen due to various reasons, that is going to cause injury to the cell and eventually will cause cell death if it is not reversed. So hypoxia, the reason it does this is because it's going to reduce aerobic oxidative respiration, and so because of that, you're then going to have a cell injury. Now, how can this happen? There's a couple of different ways this can happen. So you can have ischemia, which is just reduced blood flow, so you're no longer bringing in that oxygenated blood. You can have inadequate oxygenation of the blood, so even if you're bringing enough blood, if that blood doesn't have enough oxygen in it, that could be a problem, and that can be caused by cardiorespiratory failure. Another cause could be decreased oxygen carrying capacity of the blood. So again, you're bringing enough blood, but that blood doesn't have enough oxygen in it. So this could be caused by something like carbon monoxide poisoning or anemia. And there's a ton of different types of anemia, which we'll get into later on in subsequent chapters. And then it can also be caused from severe blood loss. So if you don't have enough blood, say you have a big injury, say you have a huge cut and it lacerates your femoral artery and you're bleeding out, well, you're not going to have enough blood in your body to be getting that oxygenated blood to whatever cells and tissues. So that can cause cell injury and eventually cell death. So those are just some examples of hypoxia or oxygen deprivation. Now, another thing that can cause cell injury are various physical agents. So this could be things like trauma, temperature, so really hot, really cold, sudden changes in atmospheric pressure, radiation, shock. And once we get into later chapters, we're going to learn how each of these, affects the cells. You can also have chemical agents or drugs, so things like glucose, salt, oxygen. So those are things that, your body normally needs, but if you have too much or too little, then that can cause cell injury. Poisons, that's kind of common sense, but things like arsenic, cyanide, those are going to injure your cells and in your tissues. Other things, carbon monoxide, we already mentioned that, asbestos, pollutants, drugs, things like alcohol or other recreational drugs, and even therapeutic drugs. So some drugs, as you probably are aware, can have really negative side effects on the body. There's also infectious agents, and there's way too many to name. We're going to talk about a lot of these later on in general and systemic pathology, but in general, you can have viruses, parasites, bacteria, and various types of fungus that can cause various types of cell injury. And each one is going to have different types of cell injury and different mechanisms. You can also have immunologic reactions, so things like autoimmune diseases, or if you have immune reactions to any external agents coming in. You can have genetic derangements. So there's a ton of these. We're going to talk about these when we get to the genetic chapter, which I believe is chapter 5. We're going to learn about some of these. You've probably already learned some of these in your embryology class. You can have nutritional imbalances, so if you have a deficiency of some sort of, vitamin or other type of nutritional thing that you need for these cells, or if you have too much of them, that can cause injury to the cells. Various types of bacteria and fungus that we already mentioned that. So all of these, right now, this is just kind of an introduction to the things that can cause cell injury, just to make you aware, and then later on in general and systemic pathology, we'll get into the individual types. So all of these are going to cause cell injury, but then what are the actual morphologic changes that you're going to see in cell injury? We talked about some of these when we talked about reversible versus irreversible cell injury. So reminder, reversible injury, here's some of the morphologic changes you can see. So remember, the whole cell is going to swell, its individual organelles are going to swell, that plasma membrane is going to start to bleb, some of the ribosomes are going to be detached from the endoplasmic reticulum, and you'll see clumping of that nuclear chromatin. So there's two main light microscopy features that you're going to be able to see when a cell is injured but it's still at that reversible stage. So the first one of these is cellular swelling. So this is going to happen when the cells are incapable of maintaining that ionic and fluid homeostasis. And importantly, this is the first manifestation for almost all forms of cell injury. So you lose those pumps in the plasma membrane, and then that's going to cause an influx of sodium followed by fluid, which can then cause the cell to swell. So this sometimes can be hard to see on light microscopy, and it's a lot easier if you kind of have some tissues to compare. But it is one of the things that you can see on just a regular light microscope for reversible cell injury. Now the second one is fatty change. So fatty change or hydropic change, this is going to be when you have lipids kind of inside the cytoplasm, so almost like an inclusion of lipid inside the actual cell itself. And this is typically going to happen when you have some sort of toxic or metabolic injury, or if you have some sort of hypoxic injury. And so as we move through the organ systems, we'll see that some organs are more likely to have this, like the liver, for example. When the liver has an injury, it is going to respond by having a lot of fatty change. That's how you can get steatosis or fatty liver. Now here, this is an example. So here, these are the kidney tubules, which you should remember from histology. So on the left, these are normal kidney tubules, and so you can see that the cells are nice and cuboidal. The cytoplasm looks normal, but this on the right, this is an early or reversible injury, so probably an ischemic injury to the kidney, and so you can see that there's some surface blebbing. You can see that they are pinker or they have increased eosinophilia, and you can see that some of them are swollen. So like this one over here, it's very pink, it's big, and it's swollen. Same thing with this one over here. So those are some reversible changes right there. Now this is showing you some liver cells where you can see that there's a lot of fat or fatty change. So over here, these are just some, let's make it a better pointer. These are just some regular-ish hepatocytes. So here, these are more regular hepatocytes. And then you can see these over here, they're huge and they're full of lipid. There's a lot of lipid in the cytoplasm. So that is fatty change. Same thing over here. So these would be just your normal hepatocytes. And then these would be examples of fatty change. So you can see the lipid within the cytoplasm. Here's another example. So here you can see these are your normal hepatocytes, and then all of these, they've got that like cloudy look that I know we've talked about in histology. So you see a lot of lipid inside the actual cell cytoplasm itself. And then if we zoom in on those, this is a very zoomed in view of those, and so you can see it's literally like little lipid droplets inside that cytoplasm. So like I mentioned, you can also see some of that increased eosinophilia, which means they're going to stain more pink. So this is because you're losing that cytoplasmic RNA, and that's what binds the hematoxylin, and the cytoplasmic proteins are becoming denatured, and that's what's binding the eosin. So you're having a lot more pink staining as these cells are injured. So we mentioned as these cells are injured, they're going to become more pink, and that means they're becoming more eosinophilic. And as this progresses to an irreversible injury, you're going to see even more of that. So this is because as a cell is irreversibly injured and starts to undergo necrosis, you're going to lose that cytoplasmic RNA, and that's what's binding the hematoxylin, which is the purple dye, and you're denaturing those cytoplasmic proteins, and those are what bind eosin. So you're having more eosin staining kind of more spread out as these are denatured, and then you're losing a lot of that purple stain. And then you're also going to have nuclear changes. So there's three main nuclear changes that you're going to see in necrotic cells as they're becoming necrotic. So the first change is going to be karyolysis, which is the basophilia of that chromatin is fading. So again, you're losing the basophilia, and these are becoming increasingly eosinophilic. Now the second change is going to be pycnosis, and pycnosis is basically shrinkage of that nucleus, and as it's shrinking, it's going to become more basophilic. So the cytoplasm itself, all of that other stuff is becoming very pink, but as the nucleus is shrinking, it's going to stain a little bit darker because it's all kind of coming together. And then the third change is going to be karyorexis. So this is when that shrunken nucleus or that pycnotic nucleus is going to undergo fragmentation. So here in this picture, this is a really good picture kind of showing you what I'm talking about. So over here on the left, you can see the karyolysis. You can see the nucleus itself is fading because the chromatin is kind of dissolving, but then as the nucleus shrinks or becomes pycnotic, that is kind of condensing, and so as it's condensing, it becomes darker or more basophilic, and then eventually that nucleus is going to fragment, and that is when it's karyorexis. So as this undergoes fragmentation, then as all of this is happening, it becomes a fully necrotic cell. So going back to those kidney tubules now, so these are the same two from the other slide. So here, these are normal kidney tubules. In the middle, this would be, this is still a reversible cell injury. The plasma membrane is still intact. They're just a little bit swollen. You can see that some of them have increased eosinophilia, but overall, this is still probably a reversible injury. Whereas now over here, you don't really see a nice plasma membrane for the cells. You can see some of the nuclei have dissolved, so like some of these don't have nuclei pretty much at all anymore over here. And then this one over here, this looks like it has kind of spread out, so this would be less basophilic, so these are more eosinophilic, so this would be your karyolysis. So basically that nucleus is kind of fragmenting, or splitting apart, and then as it shrinks, it becomes pycnotic. There's not a perfect example of this, maybe this one. And then over time, that is going to split apart, which this is a decent example of that. And then it's going to dissolve completely and be these guys over here. So this would be an example of irreversible cell injury. These are necrotic cells. These are necrotic kidney tubules. Another example is heart. So this is your cardiac muscle. So again, we see that the nuclei, you see multiple nuclei, you can see branching, you can see the intercalated disc, so think back to histo. So this is fairly normal cardiac muscle. And then here you can see, a lot of them don't even have nuclei anymore. Some of the nuclei are shrunken or pycnotic. It's hard to even recognize the nuclei because they've broken up, and you see a ton of inflammation. So all of these guys, these are inflammatory cells coming in. You see some red blood cells. So compare the nuclei over here as to these normal-ish nuclei. So those are some of the features of cell injury, both reversible and then irreversible. Now we're going to move on to the morphologic changes that you can see in necrosis, so in the different types of necrosis. So we just went over, in general, some nuclear features of necrotic cells, but now we're going to talk about the different patterns of necrosis within tissue. So this was cells. Now we're going a little bit bigger picture into the different types of tissue necrosis. So there's a lot of different types. You can have coagulative necrosis, liquefactive necrosis, coagulative necrosis, liquefactive necrosis, gangrenous necrosis. So when you have, gangrene or something that's gangrenous, that is a form or a pattern of necrosis. You can have caseous necrosis, fat necrosis, and fibrinoid necrosis. So each of these is going to have kind of a different pattern, so a histologic appearance and a gross appearance, or different morphologic changes, but they're all different forms of necrosis. So these, it's going to kind of depend on the tissue or the organ that these are in, and each type of tissue is going to respond in a little bit different way. So this is an example of coagulative necrosis. Now, coagulative necrosis is unique because the architecture of the dead tissue is going to actually be preserved for a couple days. And so typically these are going to be firm. So basically what happens is the injury is going to denature the proteins, but also it's denaturing those enzymes. And so those dead cells can't really be broken up. And so as a result, you're going to have these eosinophilic anucleate cells that kind of just sit around for a couple days, sometimes even weeks. And so this is going to be specific to a couple different types of tissue. And this is what we call an infarct, right? So grossly, an infarct looks like this. So here, this is a wedge-shaped infarct, and this is in a kidney. So here, this is normal-ish, actually not really normal, but this is kidney, and then this is this wedge-shaped infarct that's kind of like yellow tan here. And so that's what it looks like grossly. So that's what we call an infarct. Now, when you take a section of this and look at it histologically, you can still tell that this is kidney because you can see a glomerulus here. You can see, you know, some tubules, but it doesn't look really good, right? And then if you look over here, you can see that all of these, you don't really see any nuclei in any of these cells. So these are just anucleate cellular debris, really, that are just kind of sitting here. And that's because those enzymes can no longer function to kind of break up all of these cells. So they're just going to sit here. So you can see it kind of still has the architecture of what a kidney tubule probably looked like, but you don't see any nuclei. They're just kind of sitting there. So that's coagulative necrosis. Another type is the liquefactive, and this is classic for brain. So when your brain starts to die, when the brain cells start to die, they literally just turn to a liquid. So that's why it's called liquefactive necrosis. So basically, instead of coagulative necrosis where it becomes firm, this is becoming a liquid, kind of viscous mass. So this can be seen in some bacterial and fungal infections, mainly because these are going to stimulate the accumulation of leukocytes, and then that's going to cause enzymes from these cells to come out, and then they can kind of eat up the tissue and liquefy it. But classically, this is going to be found in brain, and so it's going to be usually this like viscous yellow kind of, I don't know, gross liquid, and so all of that in here, this would be like normal-ish brain. Over here you can see the white matter, the gray matter, and then all of this is an area of liquefactive necrosis. Here's another example over here. So if you, let's say, block the blood supply to that area of the brain, that tissue starts to die. It's going to liquefy into this viscous fluid, and if you take a section of that, this is what it's going to look like histologically. So histologically, you're going to just see loss of neurons, and just this formation of a clear space in the center. So here, this is just like the broken up tissue in the middle here. Next is gangrene. Oh, sorry, this is kind of blurry. But gangrenous necrosis is not really a specific pattern of cell death, but this is something that we use all the time in clinical practice. So typically you're going to hear about this when we're talking about like a leg amputation or like a toe amputation. So a good example of this is somebody is diabetic, they can have blockage of the blood flow to the extremities, and they also can have diabetic neuropathy, so sometimes they don't even realize that they have this wound on their foot or their leg, and so it can start to die. And so typically it will be some sort of loss of blood supply and necrosis. So typically that's going to be like a coagulative necrosis, like we saw that first example. So you block the blood supply and with a clot, and that's going to cause death. Now, over time, that's going to become what we call gangrenous. And so if this is something that's coming like from bacteria and brings in leukocytes to kind of get rid of that bacteria, I guess, that can be something called wet gangrene, versus this can be, this next picture is going to be dry gangrene. And so either way, it's both gangrenous, it's both gangrenous necrosis, but this would be an example of dry gangrene. So it's going to be, it's kind of like deader. So it's been there for longer. It can also kind of almost become mummified, and over time it will just get worse. And so you'll see this like dusky, we call it dusky. If you see dusky in a gross dictation, that's usually somebody who's grossing and dictating, trying to tell whoever's looking at those slides like, hey, this is gangrenous or this is necrotic. So this is a good example of that. So this would be a dusky area. It's like blackish brown. Sometimes can have this green tint like you're seeing here. So all of that is gangrenous necrosis. This example, this is like cheesy looking. So we call this caseous necrosis. So here, these are both examples of caseous necrosis. And this is classically going to be found in a tuberculosis infection or a TB infection. So we call it that because it looks cheesy, so caseous necrosis. But on histologic examination, it doesn't really have that appearance. And so histologically, what you're going to see is in the center, that necrotic area basically just looks like a bunch of fragmented or broken up cells and debris. So this debris, it's going to be just this like amorphous eosinophilic debris kind of in the middle, and then classically it's going to be surrounded by this distinct inflammatory border. And so this appearance is going to be something called a granuloma, and that's going to be important when we get to systemic path and we're talking about tuberculosis. So we'll have these caseating granulomas, and that is indicative usually of TB, especially if you're seeing it in the lungs. And that's what this is, by the way. This is a lung. Both of these are lung. You can also get fat necrosis. So fat necrosis is when it's like basically fat's response to necrosis. It can be a little bit calcified almost, and it will have this firm whitish yellow appearance kind of in the middle of fat. So this can be from trauma. So this can happen in the breast, which is, I believe, what this tissue is here. If you have some sort of trauma to the chest and it kills some of the fat cells, they're going to respond by undergoing fat necrosis and almost getting this calcified appearance. Another place this can happen is in the pancreas, because the pancreas is really fatty. So this is an example of the pancreas with focal areas of fat necrosis. So if you have any sort of cellular injury to the pancreatic cells, the exocrine cells of the pancreas, that can lead to it releasing these enzymes, and those enzymes are going to damage that fat inside the pancreas. And so when that happens, what basically happens is that's going to produce soap. So this is saponification, and that's going to appear grossly as these white, kind of almost chalky masses within the pancreas tissue. So here you can even see a duct. This is a really nice section of a pancreas. So what this one looks like histologically, you can see here some fat, you have some fibrous tissue, and you have inflammatory cells. And then sometimes, not really in this picture, but sometimes you will have some calcified areas too. And then lastly, we have fibrinoid necrosis. So I didn't really have a good gross photo of this because this is typically going to be in vessels. So it's typically going to be found in arteries, and the wall of the arteries are going to have this bright kind of circumferential pinkish eosinophilic area of necrosis, and then you're going to have a ton of inflammation. So you can see all of these inflammatory cells in here. You can see the ring of necrotic tissue. And so this is a special type of necrosis, and it's typically only going to be found in blood vessels that are having some sort of immune reactions. And so when we get to the systemic path and we start to talk about vascular pathology, we're going to see this a lot. And here's another example of the fibrinoid necrosis. So basically it just looks like fibrin. That's why it's called fibrinoid necrosis. But you can see that this was a vessel, and now it's basically almost just replaced by all of this necrotic pink tissue interspersed with inflammatory cells. And then lastly, you can have these cells that are, all these cells are dying, but typically they're going to be cleaned up. So even in an infarct, even if it sits there for a while and takes a while for it to be cleaned up, typically most of these necrotic cells and their contents are going to be cleaned up by phagocytosis or enzymatic digestions or both. But if they're not, you can get something called dystrophic calcification, and this is just those dead cells basically becoming calcified. So we'll see that in various types of necrosis later on. So in summary, we went over the morphologic alterations not only in cells but also in tissue. So we started off with reversible cell injury. So remember, reversible cell injury is going to have some cellular swelling. You might see some fatty change or hydropic change. You'll see some plasma membrane blebbing, but you don't see complete loss of integrity of that plasma membrane. You can see things like mitochondrial swelling and dilation of the endoplasmic reticulum. But overall, the cells can still reverse from that. So you can still see some increased eosinophilia, but you can also see that in necrosis. So be careful with that one. That's kind of a feature of both of these, so that's not like pathognomonic for either. But in necrosis, you'll have increased eosinophilia of the cells, but more importantly, you'll have those nuclear changes. So you'll have nuclear shrinkage, you'll have nuclear fragmentation, and then the nucleus will also just be like dissolved. You can see breakdown of the plasma membrane, so you lose that plasma membrane integrity. And then those cell contents will leak into the extracellular space and the surrounding tissue. So those are the cell morphologic alterations. Now, we then went into tissue, so we went into the patterns of tissue necrosis. And so remember that various types of tissue are going to respond differently to cell death. And so depending on the tissue and depending on the sort of injury, you may see different patterns within that tissue, both histologically and grossly. So some of the ones we went over were coagulative, gangrenous, caseous, fat, necrosis, all of those. And we'll learn more about those once we get into the organ system. So for now, just understand the basics of each.